There are several structural differences on the outer surface of bacterial cells when they are compared to the outer surface of eukaryotic cells, and the innate immune system is able to recognize those structural differences. We're going to group those differences, those structures together, and we're going to refer to them as pathogen-associated molecular patterns, or what is often shorthanded as PAMPs. All of those things that we mentioned, the thick layer of peptidoglycan in the gram-positive bacteria, and the outer membrane with its LPS in gram-negative bacteria, and the waxy coating with the glycolipids interspersed in the acid-fast bacteria. Those are all PAMPs, pathogen-associated molecular patterns that can be recognized by our immune system, our innate immune system, as being foreign. Now, there's another category of thing that gets recognized by our innate immune system, and that category is referred to as DAMPs. DAMPs refers to damage-associated molecular patterns, and DAMPs are actually molecules, chemicals, that get released from these pathogenic microbes as the cells become damaged and die off during the immune response. So as pathogenic cells die again during inflammation, they're going to start breaking apart, and they're going to release chemicals, molecules that are not found in the human body normally. These molecules will also stimulate innate immunity. These collectively are called DAMPs. Now, how exactly does the innate immune system recognize PAMPs and these DAMPs? Well, it turns out, like a lot of recognition in living organisms, it involves receptors and binding of these PAMPs and DAMPs to the receptors. There are what are called pattern recognition receptors on the surface of the cells in the innate immune system. We shorthand these as PRRs. Like most receptors, they're just proteins. They're proteins that are embedded in the surface of the membrane of certain immune cells. And the PRRs, these receptors, are able to bind to the PAMP structures and bind to the DAMP chemicals. And anytime materials bind to receptors, remember, the receptor will change shape and something will happen. Some process will be triggered. In the case of innate immunity, it's inflammation. So the PRRs that are on the surface of innate immune cells are going to trigger inflammation when they encounter PAMPs and DAMPs. Specifically, we refer to these cells that are doing this work as sentinel cells. Remember, this is the first encounter that the innate immune system cells have. So these are the sentinel cells. They're right out there on the front line, keeping watch. Now, something to know as we talk about the innate immune system. Remember, microbes are incredibly diverse, right? So all microbes don't have the exact same structural differences. We've talked about that. We know that gram-positives are different from gram-negatives. We know that they're different from those acid-fast organisms. We know that different species of gram-positives can have different structures on their cells. So it's critical for us to remember that not all pathogens have the same PAMPs. Not all pathogens have the same DAMPs. And that means that the innate immune system has to consider and respond to the most common PAMPs, the most common DAMPs. There are many, many, many PRRs on your sentinel immune cells, but some microbial structures are not recognized. There isn't a PRR for them. Now there are two images on this slide for us to look at. On the left-hand side are simply some of those structural differences that we've just talked about. Up at the top, we have a drawing of the cell membrane and the cell wall of a gram-negative bacterium, just a little piece of the cell membrane that's been cut out. In the middle, this is a representation of a sliver of a gram-positive cell with its cell membrane and its cell wall. And down at the bottom, this is a representation of an acid-fast organism with its cell membrane and its waxy coating outside. So if we start at the top with the gram-negative cell, we can see the cell membrane here. We can see the phospholipid bilayer, yeah? Then we move into the thin layer of peptidoglycan. Then we have the outer membrane that's unique to gram-negative cells. And then we have these extensions of LPS, lipopolysaccharide. Now they're showing us that there are some things embedded in this outer membrane, and that's what we mentioned a minute ago. Things like this molecule called lipid A. We don't find this in our membranes. And so the innate sentinel cells have PRRs for lipid A. And if they encounter lipid A, those PRRs are going to bind to the lipid A. If you go down to the gram-positive drawing, we can see a thick, thick layer of peptidoglycan, and again, things are embedded in it. We have this molecule called lipoteichoic acid in projections off of the wall, and then there are lipoproteins in here that are not found in human cells. There are PRRs for lipoteichoic acid and for these unique lipoproteins. There are also PRRs for mycolic acid and some of the glycolipids that are embedded in it. Now, if you go over to the right-hand side of this slide, you can see up at the top just this general category of pattern recognition receptors, or PRRs. These are protein receptors that are on the surface of the membranes of your sentinel cells, your innate immune system sentinel cells. And there are hundreds and hundreds of PRRs. They are there to respond to the most common of the PAMPs and DAMPs that occur in nature. Some of those are membrane bound, as we said, like these structures. But there are also DAMPs. There are the things that come out of cells only when they're damaged. So some of those are in the cytoplasm. Some of them are within vesicles inside the cell, and so on. There are lots and lots of PRRs, but there's not a PRR for every single structure that is on or in a pathogen. Let's talk for a minute about the sentinel cells that are such a critical component of your innate immune system. Remember, we said these sentinel cells are going to recruit leukocytes into the area where a pathogen has invaded. And leukocytes, that leuko just translates as white. So these are white blood cells. If you look up here on the upper left, this is just a drawing of the different kinds of white blood cells that you have. There are neutrophils, there are eosinophils, there are basophils, there are cells called monocytes, there are cells called lymphocytes. You have multiple types of white blood cells. And again, some of them are circulating in your bloodstream. They're in the lymph system or the lymphatic system in your body. And some of them are actually out in the tissue, protecting you. Down on the left-hand side, we have an actual light microscope image of a blood smear. And this is from a human. You can see these smaller circular structures. These are red blood cells. And the larger structures that have picked up a darker stain, these are different types of white blood cells. This is a great smear because it has several different types of white blood cells visible. You can see a neutrophil here with its very characteristic segmented nucleus. Yep, that's a nucleus. Here's a neutrophil. There's another neutrophil over here. This one over here, this is an eosinophil. Eosinophils look a lot like neutrophils, but they have these very distinct red staining granules in the cytoplasm, and you can see those in this cell right here. Basophils also look like eosinophils, but they have blue staining granules in them. You can see that over here. This quite small cell with a great big nucleus, this is a lymphocyte. And this much larger cell, this is a monocyte. So I'm not going to be asking you to tell me the different kinds of white blood cells, and I'm not going to ask you to identify white blood cells in a blood smear. That's not something you need to know for this course. But I want you to understand that part of what sentinel cells do is they send out a cry, they send out an alarm to call in the white blood cells so that the white blood cells can participate in inflammation and fight off this pathogen. So the question is, what specifically are the sentinel cells? And that brings us over to this diagram over here. The first sentinel cell is one of the white blood cells. It's the neutrophil. The second type of sentinel cell is what we call a macrophage, and it's shown on this image right here. Macrophages are actually produced by monocytes. And remember, monocytes are one of the white blood cells. Monocytes can divide and sort of mature to produce different types of cells. So the monocyte is sort of the immature version of this cell type, this cell lineage. And there are conditions, there are certain times when a monocyte will be triggered to divide and produce other types of immune cells. And those include the macrophage, a cell called a microglial cell. And if you remember from anatomy, this is a cell that's up in your nervous system. And finally, sort of a non-specific cell called a dendritic cell. A dendritic cell gets its name because it looks like it has the dendrites that you find on a neuron. Again, if you remember from anatomy, neurons have these extensions of their membranes called dendrites, and dendritic cells have those as well. So, the sentinel cells of the innate immune system include neutrophils, macrophages, dendritic cells, and cells called mast cells. And the characteristic that mast cells have is that they're full of granules that contain histamine. And you're probably aware that histamine is one of the chemicals that promotes inflammation. If you're unlucky enough to suffer with allergies, for example, you probably use antihistamines, right? You probably have used over-the-counter medications that are made of antihistamines. So let's take a look at this slide and the diagram or the drawing of some of these sentinel cells. This drawing is intended to remind us that these cells are not limited to the bloodstream or the lymphatic system. These cells are also patrolling your tissues. For example, you have macrophages circulating in your bloodstream, but you also have macrophages, for example, in your liver. These are specifically called Kupfer's cells, and they are present in the tissues of your liver to encounter and respond to any pathogens that might invade your liver. You have macrophages in your spleen. You have macrophages in your lungs. You have macrophages inside your peritoneal cavity, just the body cavity inside of you. You have macrophages all throughout your body ready to respond to pathogenic invaders. Here's a drawing of the mast cells I mentioned a few minutes ago. And here, these are examples of those dendritic cells. This is a drawing that represents the microglial cells that are the immune cells of your nervous system. And down here we have Langerhans cells. These are the types of dendritic cells that live in your skin and act as sentinel cells against invasion. It's the sentinel cells that have the PRRs, those receptors that bind to PAMPs and DAMPs from microbial invaders. And when that binding occurs, it triggers a response, and the response that it triggers is inflammation. Now, inflammation, as I'm sure you already know from probably personal experience, is a process that results in redness and heat in an area, and swelling and pain. Inflammation is no fun, but it's a really important response to help kill invading microbes. If you start at the top of this diagram, you can see that infection will trigger inflammation. So can just plain old trauma to tissue, right? Let's follow the infection path. Infection produces tissue damage. And tissue damage then, if we follow down on this central pathway, causes a couple of things to happen. It causes certain chemicals called vasoactive chemicals to be released. These are the things that are going to help increase blood flow to that area, increase the permeability of the lining of your blood vessels. Now, why would that happen? Well, remember, there's white blood cells inside your bloodstream, and they're relatively big. So we need to increase the permeability of the lining of the blood vessels so that those white blood cells can get out and travel to the site of the invasion. Tissue damage is also going to release a type of molecule called chemotactic molecules. These are being released to help, again, put the alarm out, to help call for and recruit other immune cells, particularly neutrophils. And it's the combination of these chemicals and their actions and the neutrophils arriving that's going to produce the swelling and the redness and the heat. And alongside all of this, that tissue damage is also damaging the nerves that are in that area. And those damaged nerves are going to help create the pain that we all associate with the inflammatory response. So yes, acute inflammation is no fun, but we have to remember that it's part of the innate immune response, and it's critical to help stop invasion by pathogenic microbes. Now, we also mentioned the humoral component of the innate immune system, the non-cellular component. Let's talk for a minute about that. These chemicals are actually proteins. They're relatively small proteins, and the purpose of these proteins is to facilitate communication between all of these cells. So, what kinds of proteins are these humoral chemicals? There are two that we should be familiar with. The two major types include what are called cytokines and what's known as the complement system. Now, cytokines include a variety of chemicals, and I've got some of them listed here. There are some that are called chemokines. There are some that are called interleukins. There are some called interferons. There's a type called tumor necrosis factor. All of these are called cytokine chemicals. I have interferons highlighted in this blue color because interferons are currently a subject of renewed interest during our recent pandemic. Interferons are a type of cytokine that are particularly involved in viral infection. And there's some renewed interest right now in using interferons as drugs to help fight the SARS-CoV-2 virus. Cytokines are a critical type of humoral chemical in the innate immune response, but cytokines are not benign chemicals by any means. Yes, they are critical for helping to defeat the infection, but they have all kinds of what we would think of as side effects. Cytokines are what make you feel so sick when you get infections. Cytokines, in fact, can make the disease process, the signs and symptoms of the disease process, seem really bad. For example, when you get a very high fever during infection, that's the action of cytokines. Sometimes cytokines cause such a strong immune response that the immune response actually becomes a problem in itself. There's a phenomenon that we call the cytokine storm that causes such extreme symptoms within a disease process that it's actually detrimental to the patient. So cytokines are critical, but they are not benign. They are part of what makes you feel so poorly when you're sick with an infectious disease. Now, the complement system is also a group. It's a group of proteins, about 25 or so proteins, that actually work together as a cascade. And when I use that word cascade, I mean one protein gets activated, and then that protein activates the second protein, and the second protein activates the third protein, and so on and so on and so on down the 25 protein list. And what all of these proteins are doing, the reason we call it a complement system, is they are assisting or complementing the action of antibodies. They're helping antibodies to work better. And they're also promoting inflammation. One other thing that the complement system does during an immune reaction is they help actually clean up once the immune response is over. There's a lot of debris left over after an inflammatory response. And it is the complement system that helps clean it up so that the tissue returns back to normal once inflammation is over. So the cytokines and the complement system are critical humoral components in innate immunity. Now, I have one other note on the slide, and that's that I've only mentioned a handful of the humoral chemicals that are involved. There are many, many other inflammatory mediators, molecules that are involved in inflammation. I've mentioned histamine, for example. There are chemicals called prostaglandins. There are chemicals called leukotrienes. The list goes on and on. Just want you to remember that these humoral components are just as important to innate immunity as the cellular components are. So, to summarize innate immunity, remember that first and foremost, there are physical barriers on and inside your body that help protect you against invasion by pathogenic microbes. These are truly the first line of defense, but they have a limited effectiveness, and pathogens are capable, under certain instances, of breaching those physical barriers. Any pathogens that get past those physical barriers, and again, we defined that as getting through an epithelial lining, they will activate the innate immune system. It is the PAMPs and the DAMPs that microbial organisms have that are going to bind with the PRRs on our sentinel cells and trigger inflammation. Our sentinel cells include macrophages and neutrophils, mast cells, and dendritic cells. When the PAMPs and DAMPs bind to PRRs on these cells, inflammation is going to be activated. Humoral chemicals are going to be released from our sentinel cells. Inflammation gets triggered, and it's the inflammatory response that's going to help kill and eliminate these pathogen microbes, these invaders. That inflammatory response is going to involve pain, swelling, heat, and redness. And it is this inflammatory response that will then go on to trigger the third system in our immune system, which is adaptive immunity.